Hi everyone, today I will discuss about the general embryology, which is very interesting topic to everyone because there is question in everyone's mind that where from we have come and how, and this general embryology will give you the answer. Now you know in our body there is formation of, in the adult male there is formation of the sperm, and this sperm shape is very tiny but highly mobile structure. If you see within the microscope a drop of sperm, you will see within a second 4 or 5 is going out from the visualization. And there is highly specialized ovum is formed within the female ovary, and this specialized ovum, it is highly specialized because you see there is haploid number of chromosome in sperm and the ovum, you will notice. So in our body there are diploid number of chromosome in every somatic cell. Only in the germ cells there is haploid number of chromosome. And you will see this ovum is the largest cell in the body of the female. Its diameter is about 120 mu, that means 1 centimeter and 20 millimeter is size. And this ovum is actually matured after menarche in each and every cycle. That means the completion of the second meiotic stage, and ultimately this ova is ejected. And when it is ejected, you will look. So now you see this is a picture of hysterosalpingography. It is tested for the patency of the uterine tube. You see how small the uterine canal it is in the nulliparous state, and you see how long it is, 12 centimeter the uterine tube, and the distal part you will see this is dilated and this is known as the ampulla, and it is spillage of the dye, that means the uterine tube is patent. So here ovum is picked up and in the ampullary region. So you will see here, so there is fertilization, the two specialized cell, and you see it is covered by a ring-like structure, and this is known as the zona pellucida, which prevents the entry of more sperm. So polyspermy is avoided in this way. Plus it prevents also implantation earlier. So this thing is covering whole of the ovum. So both of them fertilization occurred in this part, that means into the fallopian tube. And you see how long the fallopian tube is, and there is a very narrow, this is known as the isthmus. Why the isthmus is so narrow? Because you will see that it requires more time to pass the fertilized ovum into the uterus, because uterus requires too many times for this fertilization to make this whole of the endometrium is very soft. We think with any precious thing you will keep it in a soft portion, so uterus become more soft, and for this region there is delay in the going into the uterus. Next you will see when this two cell stage, you will notice here, you will see this is the two cell stage, not the single stage, this is the two nuclei. So you will see the two cell stage first, then one divides earlier, so then there is the three cell stage, then another, so 4 cell stage. In this way there is formation of 16 cell stage, which is known as the morula. What is the meaning of morula? Morula means mulberry like, groups of cells together that forms like that of the your grapes. In the western country the mulberry is very common. That's why they say that this is the morula or mulberry like. So this cell multiply and ultimately this cells it enters into the uterine cavity. When this cell enter into the uterine cavity, it acquires some fluid, that is the fluid inside the cavity, and it forms a cyst like structure. You see, this is the filling of the cyst. This is the, you will put in the balloon the color during the Holi. So that type of filling is known as the cyst like filling, and the whole thing is forms a multicellular blastocyst. So this is known as the blastocyst. Now this blastocyst, you will see there is differentiation of the work takes place within the cell. Some of the cells it generate nutrition. So this is known as the trophoblast. Trophos mean nutrition generating factor. Now, and the other, you will see other cells, it is more prominent and it clusters in one pole. This function is different. This produces the human being later on, and the name of this aggregation of the cell is known as the embryoblast. Now I shall discuss a little. This trophoblast multiply and ultimately this trophoblast forms an villi, that means finger like projection. That villi is not so prominent, villi is less prominent than this because I magnify this one. So there is formation of the villi, and this villi, it is so excoriative that this villi excoriates the endometrium and goes deeper and deeper within the endometrium. Suppose this is the endometrium. I will show you, this is the endometrium and this is the blastocyst. So first this blastocyst attached in this way. Ultimately this embryoblast goes deeper. Ultimately it goes more deeper. You see, and then you will see it is goes so deeper that you cannot visualize, and the whole thing is covered by the epithelium or the endothelium, you can say. So this type of implantation is known as interstitial implantation. In the human being there is interstitial implantation, so that the human embryo does not come out easily with small amount of jerk or like this. So this is not in the other animal. So after that I shall discuss about the embryoblast. Trophoblast I shall discuss when there is formation of the placenta. Now first of all, this embryo, I think this embryo which forms, that is the aggregation of the cells, and these aggregated cells ultimately unite to form a flattened disc, that is the circular disc, and this is the first form embryonic area in our body, that is known as the ectoderm. In some books it is written as the epiblast. Everything is the same. So this is the ectoderm, and next there is formation of, by the multiplication of the cells, there is formation of another cell layer that lies deep to it. So there is formation of bidermal germ layer which is circular in outline. Now gradually what happens in the ectoderm, you see this is the red color ectoderm. In this ectoderm, in the lower portion, some of the cells are specialized by the direction of the gene and the marker. You know, everything is directed, that is directed by the higher power. So this will initiate the formation of, this is known as the primitive streak, and this is capped by node that is known as the primitive node. And you see, this primitive node gives rise to formation of the cells which come in between the two layers, ectoderm and the endoderm, and that is known as the third layer, that is known as the Mesoderm, and this is known as the intraembryonic secondary Mesoderm. So within our body the three germ layers are formed. Now what will happen? I shall discuss only now the ectoderm. Why this ectoderm? From the circular part it becomes this piriform in shape, that means pear shaped, and you will see because of this primitive streak migrate the cells to the cranial aspect, not the caudal aspect. Those are the shape become this, and you will see in the center of this ectoderm, these cells become different from the peripheral cells. So the gene directed that this part will form our brain, our nervous system, which is known as the neural plate. And the red color one you will see, it is known as the surface ectoderm. So this surface ectoderm gives rise to the epidermis of the skin. You know, the skin consists of two layers. One is epidermis, that is very thin like the onion peel, and you will see the dermis is thicker, and that is developed from the Mesoderm. So this is about the ectoderm. Now you see this I have drawn here this is the neural tube. Now how this neural tube is formed in the ectoderm? Actually there is surface elevation in the region of the ectoderm in the lower portion. So this surface elevation, actually you will see it forms a groove. You will see here there is formation of surface elevation, and in between the two elevation you will find a groove here. So this is known as the neural groove. The margin of the neural groove cells, this is known as the neural crest cell. Very important, because this neural crest cell gives rise all the elementary canal nerve by which we can propel our food. Now this neural tube on the caudal aspect, when it goes to the cranial aspect, it gradually forms a vesicle like structure, and these vesicles are known as the forebrain vesicle, the midbrain vesicle, and the hindbrain vesicle. From the neural tube the spinal cord is developed. From the forebrain vesicle there is formation of our newer brain, that is the telencephalon, diencephalon. This is the formation of the midbrain, and from the hindbrain there is formation of cerebellum, pons and medulla. So this is how all our nervous system gradually develops. So this nervous system acquires some sulci and gyri when the fetus is about 3 months old. Before that, that is plain like that of toad and other things. So this is about the nervous system. Now you see, if this neural tube does not close with each other, there is formation of an opening, and this opening is known as the neuropore. And if the neuropore persists, then there is no brain formation. the patient die immediately after delivery. And you will see here in front of the forebrain vesicle, that is your brain, there lies a structure, that is the oval structure. Here ectoderm and this yellow color structure, that is known as the endoderm, is in tight contact because the mesoderm cannot go inside this, there is a gap. So this tight junction gradually disappear and forms our mouth. So this is all about the ectoderm. Next is the mesoderm, that is the middle cell layer. The mesoderm, you will see, the mesoderm has got the central part which is known as the notochordal process, and this process it develops from the primitive streak. This notochordal process does not persist in the adult life. In the adult life it is persist as a nucleus pulposus of the intervertebral disc. We have know about the disc prolapse, so that is the nucleus pulposus is prolapsed. Next, on the side you will see some elevated part of the mesoderm. And what is this elevation? This elevation constitute the paraxial mesoderm. What is this? These two are the paraxial mesoderm. Now this paraxial mesoderm, there is formation of the segmentation, and this will forms the somaid, which one can visualize on the dorsal aspect. So this is the somaid, the division, the block. Within the somaid, there is rapid growth of the somaid produces a gap, and this gap lies in this position. So this gap, it will divide the somaid into a dorsolateral part, another is the ventromedial part. Dorsolateral part form the dermomyotome. So from here the dermis of the skin and our body muscles are all developed. Now the ventromedial part, what will do? It covers the notochord and it replaces the notochord, but notochord is the induction of this sclerotome. So it forms our whole of the vertebral column, that is the spine. This is how all the things are formed by the somaid. Now lateral to the somaid there is a depression, and this depression is known as the intermediate cell mass. From this intermediate cell mass there is formation of the genitourinary system. That means in our adult life the urinary bladder, kidney, ureter, the genital system all develops from here. Now you see the next part, that is known as the lateral plate mesoderm. In the lateral plate mesoderm there is the rapid growth of the mesoderm. It will produces some cavity within. So this cavity coalesce and form a continuous cavity. This is known as the pericardio-pleuroperitoneal cavity. You see this in front of the buccopharyngeal membrane lie this pericardial cavity, and within this the primitive heart tube is developed. In front of that the mesoderm is known as septum transversum. So from this septum transversum the diaphragm is developed. How it is developed I will discuss in the next video. So there is formation of this canal, and when there is formation of this canal, when whole canal unite you see there is formation of two separate layers in the embryo. This is known as the splanchnopluric mesoderm and this is the somatopluric mesoderm. This is the somatopluric mesoderm, this is the splanchnopluric mesoderm. So somatopluric mesoderm, it gives rise to the all body muscles, and splanchno means to the viscera. That means here there is formation of the intestine. So intestinal muscle, you know intestine consists of muscle. You take the sausage, so that is the pork intestine. So within the pork intestine you will feed stuffed it with different material. So similarly you know how the muscular layer of the intestine is formed. So this is about the mesoderm. So thank you everybody, and please give me the feedback after watching the video.